Hello everyone, we are so excited to have you on board as we explore the digestion of three organic macromolecules: carbohydrates, proteins, and lipids. The organ system responsible for digesting food is the human digestive system. The human digestive system is composed of multiple organs, all helping out with digesting the food we eat into nutrients and absorbing them, which allows us to grow and repair tissues. The digestive system is composed of two types of organs: main organs, which are organs that food actually passes through. These include mouth, esophagus, stomach, small intestine, large intestine, and rectum. The digestive system also consists of accessory organs, which assist with digestion, but foods do not actually pass through them. These include salivary glands, liver, gallbladder, and pancreas. When all these organs work together, we are able to break down foods and absorb their nutrients. Let's first get a tour of the digestive system. The first part is the mouth. It is responsible for chewing food, so the surface area could be increased. Behind the mouth is the salivary glands. They are responsible for secreting certain digestive enzymes such as alpha amylase. Next, the pharynx is a flap that controls whether the air or food passageway should be opened. The food then goes through the esophagus, which serves mainly to connect the mouth to the stomach. The esophagus propels food via peristalsis. This leads to the opening of the stomach, known as the esophageal sphincter. Next comes the stomach. The stomach contains hydrochloric acid and many other digestive enzymes. It can also act as a storage for food. The stomach lining has numerous folds, which all increases the surface area so the rate of reaction can happen faster. The food exits the stomach through the pyloric sphincter and enters the small intestine. The small intestine is made up of three parts: duodenum, jejunum, and the ileum. The small intestine does most of the digestion and absorption of nutrients. The wall of the small intestine is covered with villi. Villi serves to increase the surface area of the wall so the rate of absorption could happen faster and be more efficient. After the small intestine is the large intestine. The large intestine can absorb some of the leftover nutrients such as water and minerals. Lastly, undigested food is stored in the rectum, where they stay until they come out of the anus as feces. The main organs are now finished. However, we still need to address some of the accessory organs. The liver produces bile, which is important in the emulsification of fats. Extra bile is stored in the gallbladder. The liver also plays a role in the storage of glycogen. The pancreas secretes many different digestive enzymes to help with digestion. A special organ is the appendix. The function still remains unknown, but it is believed to be the warehouse of good bacteria. Now that we've learned what each part of the digestive system does, we will next examine the digestion process of these three macromolecules. We will first start with the digestion of carbohydrates. Carbohydrates are organic molecules that are made up of sugar monomers. Polysaccharides, a big class of carbohydrates, are what we are going to focus on. Firstly, let's get clear of some vocabulary. Monosaccharides are the most basic units of carbohydrates. These are the simplest sugars, such as glucose. Disaccharides are organic compounds made of two monosaccharides, such as maltose. Polysaccharides are organic compounds made of multiple monosaccharides, all linked by glycosidic bonds. When you consume carbohydrates, they're first physically broken down by chewing, which is done by your mouth. This increases the surface area and makes it easier and faster to digest them because more locations can come in contact with digestive enzymes. Your salivary glands produce an enzyme called alpha amylase. This breaks down most polysaccharides into disaccharides through chemical digestion. The disaccharides and some remaining undigested polysaccharides enter the pharynx, which acts like a switch between the air we breathe and the food we consume, so things don't get into the wrong places. After the macromolecules travel to the stomach through esophagus by a process called peristalsis, which is the continuous contraction of muscles to move food. The entrance of the stomach is known as the esophageal sphincter. However, carbohydrates are not digested in the stomach, so they exit the stomach through the pyloric sphincter and into the small intestine, where most digestion occurs. In the first part of the intestine, called the duodenum, the pancreas secretes another type of amylase called pancreatic amylase. This enzyme breaks down the remaining undigested polysaccharides into disaccharides. Just like how alpha amylase breaks down polysaccharides into disaccharides by hydrolysis, the pancreatic amylase also executes this process by hydrolysis. Hydrolysis is the process of water breaking a bond between two saccharides, which causes them to split. At this point, all of the polysaccharides have been broken down into disaccharides. Enzymes that break down disaccharides into monosaccharides are then released by the cells of the intestinal wall. These include sucrase, maltase, and lactase. They are located in the brush border of the small intestine, and after they are released, all of the disaccharides are broken down into monosaccharides through hydrolysis, which is the process of breaking down molecules by the addition of a water molecule. Note: because the breakdown of disaccharides into monosaccharides is a spontaneous process, this reaction can help without the intervention of these three enzymes. These three enzymes only serve to speed up the reaction by lowering the activation energy. The monosaccharides are now ready for absorption. Villi are responsible for absorption. They're extensions from the intestinal border to increase the surface area of the border. By increasing the surface area, it can absorb nutrients at a faster rate as there are more locations that absorption can occur at. Villi also have microvilli, which are further extensions to further increase the surface area. Villi absorb the monosaccharides through active transport by using ATPs, and they're then transferred to the bloodstream, where they can be stored in the liver in the form of glycogen for energy. However, some carbohydrates, such as cellulose, cannot be digested. Cellulose will go through the rest of the small intestine, which consists of the jejunum and the ileum. It will then go through the large intestines untouched and eventually comes out the rectum in feces. And thus comes the end of the carbohydrates digestion journey. We will next move on to the digestion of proteins. Proteins play many different roles and they're essential to growing. Proteins, like carbohydrates, are also polymers, as they are made up of many subunits linked together. The most basic units of proteins are amino acids. There are over 20 types of amino acids, and they control the functions and the structure of proteins. Amino acids are linked together by peptide bonds to form polypeptides. A protein is made up of one or many polypeptides. After consuming foods with proteins, the foods are going to be physically broken down by chewing. This will increase the surface area so the reaction happens faster. However, this does not affect the chemical composition of proteins. They are still made up of one or more polypeptides. The proteins pass by the pharynx and travel through the esophagus, where it enters the stomach via the esophageal sphincter. The stomach lining has chief cells and parietal cells. Chief cells secrete an inactive enzyme called pepsinogen, For it to become the active enzyme pepsin, it must come in contact with the hydrochloric acid, which is produced by the parietal cells. After pepsinogen is activated, pepsin breaks proteins down into short polypeptide chains by hydrolyzing the peptide bonds. While the proteins are in the stomach, the pancreas also helps with the digestion by secreting an enzyme called endopeptidase. Endopeptidase breaks down the short polypeptide chains further down into very short polypeptide chains made up of only a few amino acids. Endopeptidase is a type of protease and consists mainly of trypsin and chymotrypsin. Remember that the stomach lining has many infolds, which increases the surface area and allows the reaction to happen much faster. When the very short amino acid chains enter the small intestine through the pyloric sphincter, the pancreas starts secreting other types of protease. These proteases break down the very short polypeptide chains into singular amino acids by targeting the polypeptide bonds. By now, all the proteins have been broken down into singular amino acids. These amino acids are then absorbed by the small intestinal wall through active transport with the use of ATPs. Finally, they are transported into the bloodstream where they can be used for different purposes, such as repairing tissues or stimulating a response. As for the proteins that are left over and have not yet been digested, they will pass through the rest of the small intestines and the large intestines, going into the rectum and eventually coming out the anus as feces. As we finish the process of protein digestion, it is important to realize that because proteins serve us so many different purposes, it is absolutely crucial for the digestion of proteins to function properly to ensure we can live and grow. Lastly, we will examine the digestion of lipids. Lipids are classified as any organic compound that is not soluble in water, meaning they mix poorly with water. Lipids also serve many different purposes. For example, phospholipids play an important role in cell membranes. In this video, we will focus on fats, which is a major type of lipids. Fats are made up of one glycerol backbone with three fatty acid tails. Fatty acid tails are hydrocarbons, meaning they are made up of only hydrogen and carbons. This gives the amphipathic property of fats, meaning that the side of glycerol is hydrophilic, while the side of fatty acid tails is hydrophobic. After fats are consumed, most of them do not break down until they reach the small intestines. This means that they travel untouched through the pharynx into the esophagus, enters the stomach through the esophageal sphincter, and leaves the stomach through the pyloric sphincter, eventually reaching the small intestines. When lipids are in the small intestine, the liver secretes a fluid called bile. Bile physically emulsifies fats, meaning it breaks down fats into smaller pieces to increase the surface area and the speed of digestion. Extra bile produced by the liver is usually stored in the gallbladder, where bile stays until the digestion process requires further bile. After the emulsification of fats is complete, the pancreas secretes an enzyme called lipase. Lipase breaks down fats into singular glycerol and singular fatty acid tails. This makes it small enough to diffuse through the cell, and thus they are absorbed into the cell wall through facilitated diffusion. Again, absorption can occur at a much faster rate due to the presence of villi and microvilli, which both increase the surface area of the small intestine. In the intestinal cell walls, the glycerols and the fatty acids are reassembled by a protein, and they now become triglycerides coated by a protein. After they are reassembled, they are transferred into the lacteal, which carries them into the bloodstream. Most fats are now absorbed, but if any remains, they will go through the rest of the small intestine for further absorption. Any remaining fats will simply go through the rest of the digestive system and come out as feces. As can be seen from the digestion process of all three macromolecules, the surface area is an important theme in digestion. The digestive system increases the surface area wherever possible. The organs that help with this includes the mouth, which increases the surface area of food by chewing, the stomach, which has numerous infolds, the liver, which uses bile to emulsify fats, and finally, the small intestine, which is filled with villi and microvilli to speed up the digestion process. We hope you've enjoyed the journey of the digestion of three macromolecules and learned something new. Thanks for watching.